get us to the second part of the discussion, in the fact that over the many years we've, become to learn of an increasing number of mechanisms that contribute to neurodegenerative diseases, and there has been a constant tendency to try to fit all of these mechanisms in the context of proteinopathies. And so I've asked Malú if she could unpack this for us and tell us whether these different mechanisms are really, pathways are fit into convergent or divergent mechanism. Thanks for accepting the challenge.
Absolutely. Thank you very much, Halal, and NSF organizers, Rice University. I'm going to hopefully stir up a little bit more controversy than Eliezer. We'll see how that goes. These are my disclosures that University of Florida requires me to share with you. And I'm going to start out just with the bottom line that Halal wanted me to mention. And this is from a really nice review from some of our colleagues, Shane Littleow, Gujan Bu, Diane Bovenkamp, which tries to summarize the common underlying unifying mechanisms for neurodegenerative diseases. And they're primarily focused on Alzheimer's and Parkinson's, but I really do think this applies to a lot of them, HD as well as ALS, PSP. And so they range from, this is the pictorial side, that's the text for the less visually oriented, aging, organelle decline, we know, lysosomes, mitochondria, they all take a dive with aging, and I'll talk a little bit more about that. Genetic contributors, and then environmental factors, which have a G by E, or gene by environment interplay. Age, diet, exercise, lifestyle choices, basically what we were told was good for the heart and anti-cancer prevention, same thing here. I'm not sure why neuroscientists took such a long time to figure that out. Heavy metals, head injury, pesticides, herbicides, solvents, metabolic and oxidative stress, a big deal, and more, you'll hear more about lipids tomorrow. Neurovascular coupling, BBB breakdown. We also think gut breach breakdown. And then neuroinflammation. And in terms of neuroinflammation, why neuroinflammation? Well, your Garanzian colleagues have spent some time studying the aging of the human immune system. And I think it's a very underappreciated phenomenon in neurodegeneration. On one hand, you have an age-related immunodeficiency, and you have an age-related inflammatory syndrome. And together, these come together to cause impaired adaptive immune responses and constitutive low-grade inflammation. And what it really is, an age-acquired autoimmunity. One of the things to notice is failure to self-tolerance with production of autoantibodies. So when you have a mutation in some of these divergent triggers, divergent genes, APP, synuclein triplication, oxidized protein, triplicates, they converge on microglia activation. This has been shown in vitro, in vivo, ex vivo, you name it, right? Now this sets off inflammatory cascades, which are bombarding neurons. Someone's got a speaker on. Okay. Bombarding neurons with apoptotic cascades, and then there's a divergence in selective neuronal population death, which is probably best explained by selective vulnerability, a concept that we don't really have a lot of time to talk about today, but we can get into in the discussion. Now, how do you get to this cascade if you don't have any of those mutations, right? So the 90% of people that have sporadic idiopathic disease, we and others propose that you get to this by these in the orange boxes, right? The systemic, chronic inflammatory system diseases. And it can be any of them, and I named a few of them in the first slide. So what is the evidence for that? Well, we know that there are a lot of biomarkers that you can follow that show you there's protein aggregates, again from the same review, synaptic markers that show synaptic decline and degeneration. Cellular markers of neurodegeneration, such as NFL, is another common one that people are tracking. There's now known to be mRNA splicing deficits, and then glial markers of neuroinflammation, both in CSF, plasma, and we've also been looking in stool and in PBMCs. And so this is the controversial part that I'm going to throw at you, and I hope we can discuss it some more. When looking at cancer reviews, this is from a myeloid-derived suppressor cell review, they talk about acute infection and trauma as something that leads to a strong activation signal, very classic myeloid activation of monocytes and neutrophils from the bone marrow. These are mobilized to resolve the threat, activate adaptive immunity, and remodel the tissue. But when you have cancer or chronic infection or sepsis, you have a weak activation signal instead, not strong enough to activate the resolving loops, and you end up with a mild but consistent production of monocytes and neutrophils. The main triggers are, though, inflammatory cytokines, and you get a pathologic myeloid cell activation, weak phagocytosis. Now, that should set off all sorts of alarms in our heads because they're not going to be cleaning the house. They're not going to be removing protein aggregates. They're going to be dropping the ball, if you will, in terms of toxic overproduction of proteins. Now, this all leads to inhibition of adaptive immunity, immune suppression, and a pathologic tissue remodeling, which in the case of cancer leads to metastases, right? So I saw this and I thought, well, I don't have to change anything in the middle. Just at the top, add inflammaging, chronic infection, metabolic syndrome, obesity, IBS, IBD, and the bottom really ends up giving you the same, an inhibition of adaptive immunity, immune suppression, neuroinflammation, and neurodegeneration. So it's a model. It's controversial, but I think it's testable. And I think what we need to recognize is that there are a lot of similarities between these processes and the way that chronic inflammation dysregulates the immune system. So if the root cause of neurodegeneration is innate immune dysfunction, if you will, then you end up having interplay with diet and lifestyle and genetics, environment, and injury. Then we should start thinking about these diseases in a different light. Not so much that we need to disaggregate or prevent the protein aggregation once it's starting, but to give some rebooting, rejuvenation to the immune cells that are aging and that are getting dysregulated with these chronic inflammatory diseases. So this is from a recent review of ours in Nature Review Immunology, where we make the case that in Parkinson's disease, that's what we need to do. We need to focus on the prodromal early stages, 10 years before the motor symptoms. And we believe that the perfect storm for Parkinson's development is aging and gene-environment interactions. Now, what do we do about the inflammation? Well, we know it's everywhere. We know it may start in the gut for some individuals. I think Patrick will talk more about this. But in the end, we know that it affects every step of the BRCA highway, if you will. And so one of the things to recognize is how are we going to identify the mechanisms of inflammation in any of these neurodegenerative diseases that are targetable without suffering collateral damage, right? And so in addition to the blood and the brain, we have a third player, and that's the gut. We know that the gut is making a lot of microbial-derived molecules, short-chain fatty acids, and that a lot of these sites are communicating in bidirectional ways with each other. And so I believe that in identifying the functional interactions between these three axes, we will identify novel targets, and it will lead to novel therapies. Now, Judy Friedman is having a cough. Okay.
Oh, I'm sorry. Please mute your. Yeah, please.
Yes. So in terms of intracellular mechanisms, I'm not a ventriloquist. In terms of intracellular mechanisms, the lysosome I mentioned before is one of those places where a party is happening. And this is from our review where we put all the genes related to Parkinson's, but we know in fact that many others related to AD are going to the lysosome. They are impinging on lysosomal function. And importantly, these have been studied in neurons a lot. And we and others have now focused on understanding how these disease-related genes are affecting the function of these organelles in immune cells. So another unifying concept is that the models available to us for studying neurodegenerative diseases are in vitro, ex vivo, and in vivo. And I encourage everybody to really try to synthesize what we know and what's going on. Non-human primates were mentioned by CHDI rep. And I think that will require a whole bunch of different models, not just one, but many, because we may not get the same kind of understanding mechanistically if we just focus on one. One of the therapeutic opportunities that we and others see is TNF, tumor necrosis factor. It's made by microglia, it's made by injured neurons, it's made by peripheral immune cells, and we know from epidemiology, and not to imply causation, that there is a good amount of evidence that people with chronic inflammatory diseases like IBD and Crohn's may have increased predisposition to Parkinson's. And if they're treated with anti-TNF therapy, then perhaps that risk goes down, right? What about for Alzheimer's and dementias? Similar story. From medical records, you see that anti-TNF blockbuster drugs appear to reduce the relative risk for dementias. And so again, it just causes you to sort of, pay attention and say, is there an opportunity here, right? And what is it? Part of the problem is that TNF doesn't come in one flavor, but in two flavors. The membrane bound is important for protection against infection and myelination. And the soluble form, which is cleaved, is pro-inflammatory, demyelinating, and neurodegenerative. So it's a two-headed monster, again, and you have to be very knowledgeable of the biology in order to target it. The drugs I just described to you are non-selective, so they're blocking both. So you get opportunistic infections, you may get demyelinating symptoms. It's not ideal. So we have been exploring targeting soluble TNF selectively in different models of neurological disease and have shown that selective neutralization of soluble TNF can give you a lot of different good things, restoring lysosomal function, insulin signaling, inflammation, both in the brain and in the periphery. And you don't have the immunosuppression because your soluble TNF arm is the one that's important and related to this. In support of this, the Alzheimer's Association funded a small immunological trial to ImmuneBio to target soluble TNF in people with MCI or AD with markers of inflammation. It's a biomarker-directed trial that lasted 12 weeks, and a few of them kept getting the drug. And all they're looking is to see, can you lower inflammation by MRI, by CSF, and what do you get? And so the answer is, in the high dose, it was an open label, you can reduce all the levels of inflammation in the CSF, you can reduce the free water, which is an indirect measure of inflammation, and there's a good correlation between those two. The Adney trial, the Adney cohort was included for reference in the blue, and that one would would have more inflammation over time. They also measured apparent fiber density, which was improved, and radial diffusivity, which is a measure of myelin health. And after 12 months of therapy, there was significant improvement. Now, by proteomics, a lot of the known usual suspects that we want to move in certain directions were moving very promisingly. So in addition to inflammation, there are many other things that are opportunities for development in neurodegeneration, such as more imaging tools, additional biomarker development, and understanding of the BBB breakdown, the gut barrier breakdown, and then again, better models that are representative. So with that, I will close and take some questions.
This was a very nice presentation, and I think it's interesting hypothesis to go after. The question I have is, Parkinson appears to have many origins, right? So is that when you say this sort of gut-dependent situation, one of the many choices? And basically you think about inflammation everywhere. Also, and now I want the real question, that is, how do you figure out who has this inflammation? How do we quantify that?
Yep.
How to choose your patients based? That's something you're going to test, because are going to start to drug a lot of people before they get sick. That
Right.
could get very crazy, right? Start to avoid every disease on people before they get sick. So I'm just curious, what's the measure?
So very insightful questions. I'm going to let Patrick address the body first, brain first, but the idea is that it could start anywhere. It could start simultaneously, and the vagus, which is one of the known or hypothesized conduits, you can have bidirectional traffic there. We know for sure in terms of signals, but also of transport of fibrils. So does it have to start somewhere for some people and others for other people, or can it start simultaneously? Maybe the gut aggregates are coming from the brain. That's entirely possible. So it is a very heterogeneous disease, but about 70% of people with Parkinson's and in the prodromal stage appear to be inflamed by stool, by plasma, and by CSF. Now, we hypothesize that in those people, the progression and potentially the pathogenesis, but we can't be ever sure about that, but the progression is probably being fueled by the inflammation, okay? We know that from preclinical models. We know that from the epidemiology, that if you stop the chronic inflammation in midlife for people with IBD, psoriasis, all that, with TNF drugs that don't get into the brain, okay, they have an apparent lower risk. So they had an intervention. They had these chronic inflammatory diseases. They had an intervention with anti-TNF drugs, and they appear to have less risk relative to the rest of us. So we need to do the clinical trials, and we need to pick people on the basis of inflammation. So in the trial that I described, they're looking for CRP that's elevated, although not quite cardiac risk, hemoglobin A1C, ApoE4, and a SED rate. And that's it, right? And so if you do that, you can capture a good percentage of the individuals. We've seen the same with ALS and AD and MSA. They're inflamed. And the question is when. So I would say that we need drugs that are not going to be immunosuppressive, that can be started at age 30, because that's when your body goes in reverse, right? Not unlike a baby aspirin, okay? So, which is not, innocuous, because we will miss the window of opportunity if we wait till age 50. I didn't have time to show the data because Halal said 10 minutes, but there's markers of inflammation that start going up, TNF receptors, TNF, at age 30, and they correlate with age.
Okay, we have time for 2 questions. We will have the panel later.
Peter. Oh, no.
So one of the perennial unsolved problems in cancer is whether the age incidence curves is really due to just the decline in the immune system as opposed to the traditional interpretation, which has to do with accumulation of mutations and other things. Is there some equivalent for maybe genetically predisposed people that you can look at age incidence curves and see if it's just consistent with decline of adaptive immune system in terms of age of incidence?
Well, I think there is some evidence from some studies. So for instance, if you have GBA carriers, heterozygous carriers that are prone to getting more Parkinson's, they appear to be more inflamed by some measures according to Wolfgang Ortel. So he claims we could enrich a trial for RBD, REM behavior disorder with GBA mutations, and do some of these immunomodulatory interventions for 18 months and see yes or no, do they progress to motor PD? That's one. There's evidence from Alzheimer's studies where ApoE4 carriers, but not ApoE3 carriers, which have chronic peripheral inflammation added onto their system, have faster cognitive decline. So I do think that neurodegenerative disease is like a cancer dance card, very much so, multiple hits, right? Genetics, environment, lifestyle, etc.
I think you just answered my question. Were you pointing at me, Halal? I can't really see well, so okay. I'm kidding. Which is there data on rate of progression being affected by these inflammatory markers as opposed to risk? Because as you know,
Yep.
wishing for wishing for pre-symptomatic trials is going to get us
I know.
no pre-symptomatic trials.
So Ron Pastuma claims that RBD is not non-motor, it's early motor, because they're flailing in their sleep, right? So it is a motor manifestation, and he truly believes that we could start there, and I second that motion.
Yeah, but I was wondering if there was data on AD progression rate where they had looked at inflammatory markers, and do they predict AD progression, or do they only predict AD onset?
They predict progression, and there's
Okay.
also for ALS. The
Okay.
fast progressors and the slow progressors. We discussed this with Jeff Rothstein not too long ago. more inflamed on the fast progressors. So I think there's evidence here and there. Yeah. Thank you.